biologic functions. His longstanding interest in mitochondrial biology goes back to his original discovery of PGC1 alpha as a master regulator of oxidative metabolism during his postdoctoral training here at the Farber. And his research continues to delve deeper into both mechanisms and physiologic contributions of mitochondrial pathways in a variety of disease settings, including diabetes, cancer, and pathologies associated with mitochondrial DNA mutations. Pere, we look forward to your talk.
Please unmute yourself. Uh-oh.
Pere, I think you're muted.
He is muted. There we go.
Okay, excellent. Yeah. Well, thank you very much, Nika, for the introduction. And it's a real great pleasure to participate and to contribute to the seminars in oncology and the Susan Surling lecture. And it's also, a nice pleasure to introduce or to present our work and to speak after Ralph, which is the work or the research I'm going to present. It's kind of good continuation along the same theme that deals with mitochondrial biology and cancer, which is one of the main processes and that our lab is interested. So, let me see. So as Ralph introduced well, to this audience, so mitochondrial organelles have an important role in cancer biology, both during tumor progression as well as in processes and resistance. And they follow and contribute the genetic evolution of the tumors, both in nuclear changes and mutations and genomic instability, as well as some mitochondrial mutation. As been alluded, the mitochondria has a DNA that follows also mutations which may be important for cancer progression. At the same time, mitochondria is responding to microenvironment interactions, and nutrient as well as environmental conditions are going to impact the mitochondria and have impact on the tumor biology. So I'm going to go briefly to general activities that take place into these organelles. So as well know that the mitochondria is the main energetic hub of the cell, and as a consequence, it harbors reactions that are either at the membrane or are inside the matrix. And these enzymes carry out important metabolic as well as energetic functions. And this includes the oxidative phosphorylation, pyruvate metabolite, and TCA saccharides included in the previous talk, but also other metabolic pathways such as amino acids, the urea cycle, fatty acids, ROS production, and then heme biosynthesis and redox reaction iron sulfur, and another pathway which is important for the redox control, which is 1-carbon metabolism. In terms of the mitochondrial biology, there is important cellular processes that have implications in cancer biology that includes the mitochondrial DNA replication that controls the mitochondrial DNA amounts. Also, all the dogmas of the cell biology as replication, transcription, and translation occurs inside these organelles that derives from bacteria. And also there is processes just the mitochondrial UPR that responds to protein folding responses, and also other proteins involved in the dynamics, fusion and fission proteins, other proteins and complexes involved in crystal biogenesis, and all the lipid transport and mitochondrial import that shape the mitochondrial protein and function. As far as cellular processes that these mitochondrial organelles contributes to the cell, and as a consequence to the organism organism in this case are, mitochondria is, harbors, important cell death pathways, which are important then for the survival. It also contributes metabolically to the proliferation as well as the growth and the cell differentiation process, redox. And an important aspect that has important consequences in tumor biology are the inflammatory, senescence, and immune processes that at the end I'm going to discuss. As Ralph, has alluded, the mitochondria has been also thought to be targeted in cancer. And as a consequence, I just have listed in this case several targets of the mitochondria that involve proteins involved in glucose metabolism, as well as components at the phosphorylation, such as complex 1 and complex 3 and complex 4, and with mixed success, which probably deals with the mitochondrial heterogeneity that happen in tumors, as well as there are other compounds that perhaps not as specific have been brought to the cancer medicine and have, with mixed success in tumors, and that might depend because we don't know exactly what are the targets. More recently, work here in this area, coming from the Broad Institute, that have generated this cancer dependency map. There are specific selectivities, particularly on mitochondrial ribosomal protein, that are important for translation of these mitochondrial proteins, or in hotspot genes, and certain tumors seem to be highly or more dependent than others, and that might provide some vulnerabilities that can be explored together in some clinical studies. So I think the mitochondrial regulation, as we know from classical textbooks in cell biology, that occurs in normal cells are probably, at least at the first look, not that different in cancer cells. So the oncogenic driving mutations, I think, shape and contribute to different functions of the mitochondria. But as far as we know, as specific regulations, probably the mitochondrial biology that happens in these tumor cells follow the general principles of the mitochondrial biology that have been discovered in many different studies by different authors. And you want to look, what control the mitochondrial abundance in these cells. That might be dependent on the cell lineage specific, and that's again have been discussed a little bit in the previous talk. But one thing, or at least one component, perhaps related to the cell lineage, that is going to control this abundance is the differentiation state. As happens in normal cells, usually cells that that non-tumor and proliferate, they usually have less mitochondrial abundance, but as cells go through the differentiation process, they try to accumulate these organelles and try to be more dependent on oxidative phosphorylation. As an example, in tissues, the tissue that tend to be more susceptible to mitochondrial dysfunction tend to be the more differentiated cells, such as the neurons, the cardiomyocyte, or skeletal muscle cells that have these main pathologies in mitochondrial diseases. So perhaps the same kind of regulation of mitochondrial abundance occurs, at least as general principle, and there are exceptions, I'm sure, that can be discussed, and there are probably some different ways that mitochondrial abundance can be obtained. And the response to the mitochondrial mutations could be also similar. And then the mitochondrial adaptations, when mitochondrial is inhibited or depressed, that might be important for conditions of resistance. So in this lecture, I'm going to divide in 3 different chapters. So on 4 different chapters, sorry, and every different chapter contains important consequence for tumor biology. The first part I'm going to discuss is going to be the mitochondrial abundance and the cancer cell type that is mainly or can be mainly related to the survival. So the way I think is being set up by different studies, at least from normal cells, and at least in certain type of tumors. So usually tumors that tend to be more differentiated, they tend to have more mitochondria. More differentiated means that the cell lineage of the tumor perhaps express more cell markers which are originating from the tumor that the tissue is being formed. So just for example, in melanoma, that is one of the tumors that I'm going to discuss, like the MITF and some of the pigmentation pathways, is what defines, or what we might call, the differentiated state. And then the undifferentiated state, at least in general, I mean, it can be, I think, cases, but tend to be with lower mitochondrial abundance. And that's, we try to investigate this in the context of melanoma. And the way we start these experiments, we're looking at one particular coactivator that is responsible or causative of mitochondrial biogenesis. As Nick alluded, so this coactivator was discovered here at the Farber in the lab of Bruce Spiegelman, and many different work has put this coactivator, at least in healthy tissues or in normal tissues, in the control of mitochondrial biogenesis. And one important thing, at least in these non-tumor studies, is that the activation of PGC1 alpha and mitochondrial biogenesis occur through many of these different transcription factors. And the activation of these transcription factors have two programs at the same time. One is mitochondrial biogenesis, but at the same time, there is a ROS detoxification pathway that mitigates or tries to decrease the oxidative state in terms of reactive oxygen species of these cells. So we probe certain or different tumors, and one of the tumors that we probe was melanoma. And these melanomas that we probe, it was approximately like 10% of the total human melanomas that express highly of this transcriptional coactivator. And this is also in melanoma cell lines or in short-term melanoma cultures. And as predictive, based on the studies in normal tissues, this expression correlated with most of the oxidative phosphorylations and mitochondrial markers. And in these different cell lines, it was clear that inactivation of this gene in these high PGC1 alpha melanomas and high mitochondria had compromised survival, and there was strong decreases in the cell number, which was not observed in tumors or cell lines that have not increased in PGC1 alpha. Another vulnerability that these tumors had is that when we deleted a component of PGC1 alpha, which is the ER alpha, these high mitochondria, high PGC1 alpha tumors were very sensitive to the depletion of this ER alpha. And then in these two different cell lines, the tumor growth was decreased. At the same time, these tumors were having these defects on mitochondrial oxidative phosphorylation. One, I think, characteristic that this tumor had was an increase in oxidative stress. The ROS levels, as a consequence of the depletion of PGC1 alpha and perhaps some decrease in this mitochondrial number, there was a burst in oxidative stress. And as a consequence, if we treat these tumors with a ROS inducer, such as the piperlongumine, then these tumors had basically no growth in xenograft models. So that means that, at least in terms of the mitochondrial abundance in these cell lineage melanomas, the mitochondrial abundance and activity was required for the growth and survival, and at the same time was more sensitive to oxidative stress. And I'm going to go back, at some point into these different features. So the second chapter I'm going to move now, and also has been alluded a little bit, at least in abundance in the previous talk, is that mitochondrial mutations in cancer cells. So the mitochondria has this unique mitochondrial DNA, which is circular, that encodes basically certain proteins which are part of the oxidative phosphorylation system in different complex 1, the complex 3, 4, and 5, and at the same time encodes the tRNAs, which are responsible for the mitochondrial translation. This, I think, is important, at least, or can be important, and has not been fully studied for example, in conditions of the chromoform renal cell carcinoma, where they appear to have, in consequence, at least for some lineage of p53 deletions, are these ND5 deletions. And these tumors, as a consequence of this ND5 deletion, will completely have an inactive complex 1 activity. So in the context of the mitochondrial disease mutations, we have studied some cancer cell lines that have mutations in complex 1. And we have asked the question, what kind of vulnerabilities can have these cells? Or in other words, what kind of other genes can promote the survival of these cells? And we use CRISPR gain of function screens to identify these genes. And one characteristic of these cell lines that have these mitochondrial mutations is that they are not they cannot grow in nutrient stress conditions or in galactose. So mitochondria is required in these conditions. If you have mutations in this, the complex 1, these cells are going to die. So one of the hits that we got in these tumor cells that have deletions or are deficient in complex 1 activity was the malic enzyme 1, which was expression of this enzyme was rescuing this complex 1 mutation. So the malic enzyme is a cytosolic enzyme which is responsible for generating NADPH and is tightly connected to the TCA cycle in a way that when the TCA cycle functions, then the citrate can be derived to the cytoplasm and used for lipogenic activity, and part of it can be used for the malic enzyme 1 activity, and in this case, it generates NADPH. So it functions, at least in this case, a kind of antioxidant, an antioxidant gene. And that might be important, at least in some cases, because Alec Kimmelman several years ago found that the KRAS human pancreatic ductal adenocarcinomas were somehow sensitive to this enzyme and were requiring this enzyme for survival. So the reason why this enzyme can be important in this context is because of the NADPH productions. And what I'm getting is that sometimes, or at least in certain conditions, defects in the oxidative phosphorylation, in this case on complex 1, what is altering is all the redox state and then NADPH production, affecting the vulnerability of the survival in response to oxidative stress. And that brings to what is important, or what are the pathways that generate NADPH, and that has been somehow very worked out by Rabinovich at Princeton, and at least in the cancer cell and in other cell types. And there is contribution from the 1 carbon metabolism, the malic enzyme, in this case, both cytosolic and mitochondrial, and then the pentose phosphate pathway. So in these conditions, we think that the malic enzyme can provide some survival benefits to cancer cells that may have mutations in complex 1. So we characterize these cells, these cancer cells with mutations in complex 1. And when they are submitted to conditions of glucose restrictions, these cells are very sensitive to cell death. But when the malic enzyme is overproduced in these cells, then the survival of these cancer cells can be somehow immortalizing the survival of these cells. And at the molecular level or mechanistic level, how then this complex 1 become sensitive to the malic enzyme, or how the malic enzyme 1 can rescue survival of these cells. So our model and our findings that I'm discussing here, or what we observed, was that in wild type cells or cancer cells that don't have complex 1 mutations, so when they are forced to glucose restriction, then in this case the pentose phosphate pathway is not producing NADPH because we are limiting the amount of glucose. So in this case, the mitochondria activates 1 survival pathway, which is the 1 carbon metabolism that derived from serine, and in this case it generates sufficient NADPH that maintains the survival of these cells. So on the other hand, if you have a complex 1 mutation, and that brings me to perhaps these renal cancer cells that they have these ND5 deletions, or any particular tumor cell that may undergo through complex 1 mutations. In this case, they cannot generate in these conditions NADPH in the cytosolic. And this case, because of the mitochondrial complex 1 mutation, it cannot generate sufficient NADPH. So in this case, are basically relying on having sufficient malic enzyme 1 activity to generate sufficient NADPH that maintain the survival. That might become that in certain cancer cells with these complex 1 mutations, they may have vulnerabilities to this malic enzyme or other pentose phosphate pathways that could be used. And that might be also illustrated in condition in non-tumor cell, like the red blood cells, that they lack mitochondria. So they are very highly sensitive to oxidative stress and highly sensitive to mutations in enzymes that produces NADPH. So I'm moving to now the next chapter in this mitochondrial biology, and I'm going to discuss some recent work in our lab that define what's the role or what contribution can have mitochondria to adaptations in tumor progressions, and at the same time, what is the role or what kind of role can the mitochondria has in response to drug-targeted therapy. So in this case, I'm going to see, like in our melanoma model, what we have observed is that there can be a transition between these differentiated high MITF, high PGC1 alpha, that they move to undifferentiated melanomas that have less mitochondria. And in this case, they tend to depend less on oxidative phosphorylation. At least it's been a decline or less abundance. And in general, when we see this transition, we see increased oxidative stress. So when they lose these cell lineage markers, including PGC1 alpha, we see an increase in oxidative stress. So that's why we think it might be more sensitive to piperlongumine. And in general, and I don't know at what step these things, we see that these cells have more invasion and metastasis, at least in some models. And one important thing that we have found is that in this newer state, when they are more undifferentiated, they try to produce more integrin and plasma membrane proteins, and at the same time that generates these new vulnerabilities. So in other words, the blockage of some mitochondrial pathways or mitochondrial regulators in these high melanoma cells, then they transition to another state which tend to be more resistant, but they're appearing some more new vulnerabilities that can be tractable perhaps. So in terms of the numerous progressions, is that getting back to the studies that I presented in the first chapter, while we were observing that at least in the short term, the loss of PGC1 alpha and this mitochondrial program in melanomas was kind of transient. So at the beginning, it was a strong suppression of cell growth and survival. But after 2 or 3 weeks, then we were trying to appear that the rate of these cells were trying to be at the same time similar. So to some extent, what we observe is that it was the classical shift that is being shown in yeast biology, for example, is that when you inhibit the mitochondria, then the glycolytic pathway turns on. And at least in our system, one of the pathway that was turning on and maintaining the survival of these melanoma cells was HIF1 alpha, this hypoxia-regulated factor. And consistent with this activation, the glycolytic pathways were activated, and these cells were able to survive both in suspension as well in the dish and in the xenograft. So one characteristic thing that we have observed, and not only in tumor cells, but in other cells, like in this mitochondrial disease mutant cell and in other cells, as soon as when we perturb the mitochondria, when we make cells that they have deficient of mitochondria, these cells respond by increasing metastatic programs, such as programs in the integrin pathway, the focal adhesion pathway, the adherent junction, TGF-beta pathway. It's a kind of resistant state that maintains survival even in suspension. And interestingly, and we don't know what stage these cells, so these cells have more migration and invasion, where they're being tail vein injected into the mice. So these high differentiated cells, they tend to be less, very less metastatic. But when we decrease PGC1 alpha and consistent with an increase in this integrin pathway, these cells become quite invasive. And we see this in tail vein model and then in the orthotopic xenograft models, in that we see massive invasion in this case. So we try to model now these resistance states, or this low PGC1 alpha, if it has any implications in human melanoma patients. And we collaborate with Hans and trying to look the expression of PGC1 alpha in melanoma patients that have undergone through the BRAF and treatment. And what we compare is that what happened to the PGC1 alpha expression in clinical specimens that before treatment and after treatment, when these tumors are resistant to the BRAF inhibitor therapy. And we observe in that, so when we compare that, most of the melanomas that pre-treatment were having high PGC1 alpha, at the end of the post-treatment that or on treatment, then these melanoma, they try to lose over the BRAF therapy the expression of PGC1 alpha, and that correlated with survival of these patients. So when they transit from the pre-treatment from high PGC1 alpha to low PGC1 alpha, it was an increase in death of these patients, and tumors that were not going through this transition, that they maintain the expression of PGC1 alpha, they try to, or they survive longer. So we try to model this in culture and in different melanoma cell lines to try to define perhaps some vulnerabilities of these cells. And we went to different melanoma cell lines in which they were become resistant or adaptive to the BRAF treatment. And consistent with previous work, the early response to this drug-targeted therapy, the BRAF and even MEK inhibitor, in both cases, we see an increase on PGC1 alpha and mitochondria. There is a correlation on that. But then over time, most of the cell types, the cell-resistant cells, were losing PGC1 alpha. And at a certain times, then they were an increase then on cell number, but PGC1 alpha remained suppressed. And we think that this suppression tend to be epigenetically. So there is H3K27 that basically is responsible for this suppression. But our interest was to try to identify vulnerability. So what happened to these cells that now have become resistant to this drug-targeted therapy? And we perform a metabolic screening, looking at metabolic drugs that were affecting the cell growth. And the top drug that was killing preferentially these resistant cells were statins, these cholesterol lowering drugs. And we were very interested because these are clinical approved drugs, and tried to investigate why these melanoma cells that become resistant to the BRAF and MEK inhibitor treatment, they are sensitive to statins. And the sensitivity was dramatically almost more than 8 or 9-fold in these cells. That could be mimicked by the deficient of PGC1 alpha, that these cells, if we remove just PGC1 alpha, can provoke the sensitivity to a statin. And in the tumor xenograft, we saw similar effects. So I'm going to have a brief summary of this result because there is a lot of mechanistic details here, but it turns out that the sensitivity of these melanoma cells, which are resistant to the BRAF, is not depending on the cholesterol branch of the statin effects. So there is the statin effects on cell growth, on cell survival in this case, is working through a RAP proteins, which are in the geranyl pyrophosphate or prenylation pathways that have to deal with trafficking of vesicles to the plasma membrane. In fact, when we compare the BRAF resistant and parental cells, the main cluster of proteins that were upregulated were plasma membranes. So it's in that when we suppress PGC1 alpha that induces this de-differentiation of these melanoma cells, the cell respond by increasing and upregulated the same pathways that we observe in the depletion of PGC1 alpha. So there's a very good correlation. And we think that these plasma membrane proteins and integrins and the cell addition is what maintained its cell survival. But why these cells were sensitive to these RAPs? It turns out, and that's probably my favorite slide of this lecture, is that that reflects a proteomic signature of a parental cell line, melanoma, that is resistant to a statin. If you look at the protein, this is like a fingerprint of the proteomic of this cancer cell, that there is almost one single cell that changed dramatically, which is the HMG CoA reductase that, as predicted, is the target of the statins in the cholesterol pathway. But if you look at a sensitive cell, look at the proteomic signature, a lot of proteins move to the right and left. So it's like a bullet that basically destroys this cancer cell, kind of displacing the proteins up and down. If we look at the proteomic then signature, that what are these proteins? These are proteins involved in the chromosome segregation, in DNA damage, in cell death, which are basically activated by statins. This can be just rescued by one single metabolite, the GGPBP, and then we can rescue this sensitivity or resistance by just expressing two different RAPs, the RAP6B and RAP27A, that we found that just ectopic expression of these two proteins rescue the survival of these proteins. And this correlates with integrin and FAC signaling. So when we have resistant cells, our statin strongly suppress FAC signaling, and that can be mimicked by PGC1 alpha depletion, so exactly the same. So deleting the single gene, and then by overexpressing the RAP6B and RAP27A, we rescue this expression. So similar effects in the resistance. And at the more cell survival, we see that these cells are not surviving in cell suspension or the dish, so there is no growth or survival. And then just expressing these two proteins, we rescue the survival of these two proteins in this case. So I think what we have identified, at least in this case, is that when we switch this differentiation program, that at least in this particular case coincides with changes in mitochondrial abundance, there is an increase in oxidative stress. Cells become more vulnerable to ROS inducers. There is an increase in plasma remodeling and invasive states, which is the integrin and FAC signaling. But at the same time, appears that these resistant cells have new metabolic vulnerabilities, in this case to the statins, which is independent of the cholesterol pathway and more depending on the prenylation pathway that provides some vulnerability to these cells. And in this case, there is another vulnerability that I have not time to talk, that in this differentiated cell, they also become very sensitive to the ferroptotic pathway. So I'm just going to spend like 2 slides on the mitochondrial immunogenicity. I'm sure the next talk is going to provide more insights in different aspects of that. But it's in that the same pathway that we have working in other tissues and in cells, such as the PGC1 alpha and mitochondrial, abundance seems to also play a role in immune cells. In this case, we have most of the chapters have been talked about the tumor cell per se, but there is an important component of the mitochondrial biology in these immune cells. And in this case, in two different aspects, this coactivator has been to be important to maintain the metabolic activity in the tumor microenvironment in these T cells and to maintain the tumor survival. But also there is effort from different groups in trying to manipulate this coactivator and mitochondrial biogenesis and activity in the T cell therapy. And from our side, we have been lately interested in trying to investigate and move into what's the role of mitochondrial immunogenicity. And I'm not going to have a lot of data here, but just one of the experiments that we have done, we have used these mouse melanoma cell lines that we have inhibited the mitochondrial complex one, and using the inhibitor that Ralph has alluded that has a strong effects in his tumors affecting the cell growth. In our mouse models, the tumor, when we look at the activity in the newt, has some effect, but is not statistically significant. The high statistically significant effect that we see is that when we have an immunocompetent competent mice, and we think that provides at least perhaps some vulnerability, and we have some additional spread. I'm going to present that affects some immune pathways that may also contribute to the tumor growth, and that goes along that these the mitochondria, in addition to this complex one in the immune cells, the mitochondria itself, because of the bacterial origin, it has many impact on the mitochondrial immunogenicity coming from the mitochondrial DNA, the formyl peptides on translation, and also harbors most of the inflammasome that can be important then in interferon signaling. So that's a conclusion slide. So I have, I think, provided some perhaps principles, in some case can be maybe tangential to some of the programs of mitochondrial biology and cancer, but I think I have touched, different aspects of the mitochondria in abundance, mutations and vulnerabilities, adaptation, resistance, and like immunogenicity that I think can provide some newer guides and newer foundations to study more in depth the role of mitochondrial biology in tumor progression, not only in the tumor, but also in the immune system, and also in the communication between the mitochondria and the tumor, and the interactions with the immune cells. With this, I'm going to close the lecture, and then to acknowledge people in the lab that have been involved in this cancer mitochondrial biology, particularly Chilu that started most of this work, but also Jason that has done most of the work on statins and in the immune. And then our collaborators in melanoma, longest time collaborator, Hans Winlot. Then with Steve, we have done most of the proteomics, and some of the immune projects that have had a chance basically to introduce a lot. We collaborate with Kai in our institute, and some of the initial work on the CRISPR screens and identification of melanoma that was done from these folks at the Broad Institute. And this is some of the funding that we have here at the Dana-Farber from the different NIH institutes and the internal programs at the Farber and the Department of Defense and some other foundations. And I thank again to Nika and the organizers of the thrilling talk for giving the opportunity to present this work. Thank you.
Thank you, Pere, for a great talk. We have a few questions. I'm going to start with one that asks or discusses that PTC1 alpha function is contradicted in different studies. It stimulates cancer metastasis in breast cancer, but suppresses metastasis in prostate cancer. Do you think PGC1 alpha is an oncogene or a tumor suppressor gene?
So that's a good question. So I think from our work, mainly tend to be the survival gene, at least and it tends to follow more the differentiated state. The more I think you have the PGC1 alpha, the cells are more survival, and that fits with all the biology and work that has been known in non-tumor cells, that PGC1 is protective and somehow anti-inflammatory or anti-ROS, or protects against oxidative stress. So now in tumors, I think some of these studies I think are problematic in a way that they are very, I wouldn't say is not very common that PGC1 alpha is highly abundant or highly expressed in a lot of tumors. So I think melanomas is one of the examples, and it might be certain tumors that it may have, high expressions. So I wouldn't say that this is general in all the tumors that they have this high expression. It could be that sometimes that MYC that tends to favor the mitochondrial biogenesis can work with other factors. So that's what I would say. To the question of whether then PGC1 alpha could be then in some cases controversial, I think the way I see is that first of all, the low expression can be problematic then, to analyze or technically to probe the PGC1 alpha. But in some other cases, it's a very highly inducible protein that it changes quickly, and the transiently and adaptability of the cancer cell is it can reprogram quite easily to different states. So I think that in our case, you touch, you decrease, you get transient decrease, but then it goes back. It's similar that happened with the drug target therapy. So it goes up, but suddenly the cell can suppress and then move on to a different pathway.
Mm. Another question relates to EMT. The person asked, EMT is highly related to metastasis and may induce mitochondrial dysfunction. On the other hand, your results seem to suggest that mitochondrial dysfunction can cause metastasis. My question is about the rational causality between mitochondrial dysfunction and metastasis.
Yeah, so I would be cautious on that. So what we present, at least with the PGC1 alpha, it seems that the association with mitochondria and metastasis is not as straightforward. So decreases PGC1 alpha, you see decreased mitochondria, but at the same time, at least there is another transcriptional pathway that links more with TCF transcription factors and the YAP pathway that have published that likely independent of the mitochondrial effects can drive the PGC1 alpha effects on metastasis and invasion. So I think that's, I'll be a little bit cautious on that. On the other hand, what we have seen from mitochondrial mutant cells is that these cells survive better in suspension, at least in normal conditions. And there is, I think, a signaling program from the mitochondria to the secretion pathway that tend to be important for survival. To what extent this is important in the tumor types or in what kind of a scenario that is important, we don't know. I think, we may study.
We have a question from Ralph asking if the CRISPR screen provided any insight into where the malate for malic enzyme comes from. Did components of the malate aspartate shuttle or the citrate malate shuttle also score in the screen?
No, I mean, at least they were not sufficient by gain of expression to score in this screen. I mean, the other components were related to the ROS pathway, thyroidauxins and peroxyredoxins. So in the other one, we might, or at least in some cases, mitochondrial IDH, but that was not from the screen. That was after we probe some of the contribution of the IDH and TCA on an ADPH production, but that was not from the screen.
Bush Wiggelman asked if can the tumor vulnerability be exploited by inhibiting OXPHOS through inhibition of ERRs?
Yes. So actually, they've been certain companies interested in targeting ER alpha antagonist to the tumors. So one of the main issues, and I'm sure from the previous talk, can be at least indicated on that, is that, and at the end with the immune regulation, I tried to emphasize this, and that was one of the problems with these inhibitors, is that if one inhibits the mitochondria in the tumor, also inhibits the mitochondria in the immune cells, and T cells tend to be particularly vulnerable to that. So that's been some, I think, caution, at least in some of these companies, to move forward these ER alpha antagonists that have been used. From our studies, it was clear that there was a clear dependency, depending on the melanoma type, on ER alpha. But at the same time, there is work in immunocell that ER alpha is important for T cell function. So one may take into consideration that.
We have a question about how you characterize differentiated and de-differentiated mitochondria in cancer cell. Is this based on aggressive nature of cancer cells, differentiate mitochondria cancer cells, or de-differentiated mitochondria cancer cells?
No, I think, I'm not that knowledge on other tumor types, and we have been mainly most of our work has been in melanomas, and in melanoma it's clear that the differentiated ones are clearly MITF positive, which MITF is the oncogene, and that drives the differentiation, and then that's linked also to a lot of the pigmentation pathway. And at the same time, you have the PGC1 alpha that, at least in a subset of the MITF, you get also these mitochondria ones. So I think in this case, in the melanoma, I think it's quite clear how to separate that. In other tumors, I would say perhaps in breast, one can do more of the ER transcription factors, or some other cell lineage specific factors that can have more identities of the original tumor type or tissue type.
Another question is, the person is wondering why cancer cells with low amount of mitochondria show tendency to metastasize. These cells seem to grow slowly in primary sites, as exemplified by PGC1 knockdown in xenograft studies. How do you explain low growth advantage makes cancer cell easily metastasize? Thank you.
Yeah, that's a great question. So I think maybe I didn't make myself clear on this slide, and I think because it was temporally different during the presentation, is that I would say that the cell growth is temporal, like in the acute ways, or in several weeks when you remove PGC1 alpha, the cells kind of collapse. But after several weeks, it's cell rescue, and when they rescue, they have the same amount of cell growth, at least, without rescuing the mitochondria, without rescuing PGC1 alpha. And
then, as a probably last question, we have more of a broad, perhaps conceptual question as well. How different are metabolic and mitochondrial abnormalities between what happens in cancer and what happens in aging?
Yeah, I think that's a great question. I think that might depend maybe on or my intuition, and again, so I would say just intuition, and I don't know how much rational. Sure, there is not much of scientific data. What I would say is that there are a couple of pathways that may be kind of similar, although maybe cancer is maybe more strong or maybe more aggressive in this case. So in the aging, we get all of these senescent inflammatory pathways. So in this case, one might consider that tumors with high degree of inflammation, or hot tumors, might be similar to that. At the same time, it could be that similar, tumors that have, high or elevated rates of mutations in the mitochondria they might generate, at least some vulnerabilities, but also may have some of the aging phenotypes. So that's from the tumor perspective. I mean, another angle is like other groups are focusing even here and other places is that is the opposite. And you have mentioned at some point is how then the aging is affecting the tumor per se. But that's completely speculation that we don't have clear picture.
Yes. Perry, this was great discussion and a wonderful presentation. Thank you again.
Thank you, Inka.
And we move on to our last